Good afternoon, everyone. I am Aaron Pollock, Program Head for Pathology and the Laboratory Medicine Program at Cancer. On behalf of Cancer Care Ontario, the Partnership Against Cancer, and the Canadian Association of Pathologists, I'd like to welcome everyone to today's checklist education session on cancer biomarker reporting templates. As our speaker may get underway, I would like to take care of a few housekeeping items. The session is being recorded and will be made available to all participants via email links once the recording becomes available. The live presentation and the recorded presentation are eligible for CME credits. Information for obtaining credits was provided in the notice that previously distributed. CME certificates for each of the CAP checklist education sessions will only be issued for 1 month from the presentation date. Please refer to the session notes for the exact deadline. Everyone's line should have been automatically muted for today's presentation. If the presentation is clear, I would ask centers to also mute their lines directly. Which number of participants, and maybe some difficulty troubleshooting WebEx issues directly as part of this call. If you're having difficulties, contact the WebEx support line directly. Number has been supplied in the email this presentation. We encourage you to submit questions anytime during the presentation using the chat feature. The documentation previously distributed provides instructions for using the WebEx chat window. Question and answer portion of the presentation. In order to avoid question collision, a CCO staff member will pose the submitted questions on your behalf as long as time permits and the order in which they appear. So please include the following information: your institution, your name, and your question. We're very fortunate today to have Dr. Dr. Lazar for our presentation. Dr. Lazar was born in Santa Ana, California. Obtaining dual degrees in philosophy and political science at Boston College, he entered the NIH-sponsored medical scientist training program at UT Southwestern in Dallas. He did an atomic pathology residency at Brigham and Women's, Harvard Medical School, and fellowships in soft tissue pathology with Cliff Fletcher and dermatology pathology. Dr. Lazar is currently an associate professor at the University of Texas MD Anderson Cancer Center. His clinical work focuses on diagnosis of bone soft tissue tumors and soft tumor molecular testing. He's focused on next-generation-based sequencing for therapeutic selection and triage to clinical trials, and has a particular focus on melanoma. Founding director of the ACGME accredited soft tissue pathology fellowship program, he's the next generation of bone and soft tissue pathologist and translational researcher. His research has centered on the molecular characteristics of melanoma and many sarcomas, correlation with clinical outcome, and searching for novel therapeutic interventions. His work is published in more than 250 peer-reviewed manuscripts, book chapters, and edited books. As a senior associate editor of Laboratory Investigation and as a member of additional editorial boards, Dr. Lazar is deeply committed to the peer review and publishing process. Leadership positions with the American Joint Committee on Cancer for both melanoma and sarcoma. With CAP, Dr. Lazar is currently on the Molecular Oncology Committee and was previously on the Cancer Committee of the Cancer Biomarker Reporting Committee. CC protocols for defined pathology reporting on tumors, including molecular diagnostics and proficiency testing. Participated in the 2013 World Health Organization classification system for bone and soft tissue tumors. As a result of this collaborative work with numerous colleagues, 3 systems for sarcoma are now completely aligned for diagnosis, reporting, and staging of sarcomas used and adapted worldwide. I believe with all these qualifications, we're very fortunate to have Dr. Lazar, who would be very familiar with the CAP protocols and biomarker checklists. And without further ado, I'd like to introduce Dr. Lazar to give today's talk on cancer biomarker reporting templates.
Hi, am I on now? Yes, you are, and you can share your desktop now.
Great, yeah. So it looks like I just got control, so now I'm going to put first slide up. Is that visible now? First slide visible?
Dr. Lazar, if you could please click on share your desktop, and then we'll be able to see.
Okay, yeah, sorry. Let's see. I'm looking for it. It is share desktop. Okay. And I will share screen 1, which is where the presentation will be. Okay. Okay, everything went blank, and now it's coming back up. And
maximum tumor.
Okay. Now is my first slide available or visible?
Yes.
Okay. All right. So okay. Sorry about that, guys. We couldn't actually test this until we were actually running with it live, and so that's the but now I think we're ready to go. So what we're gonna talk about today, we're getting started a little bit late, but what we'll talk about for the next hour or a little bit less are the CAMP cancer biomarker reporting templates. I want to talk to you a little bit about what they are and how they're intended to be used. So the next slide is supposed to be my disclosures. This is my slide saying I have possibly relevant disclosures that are going to be on the next slide, and then saying that there will be some discussion of off-label pharmaceutical use, but it's only within the context of a formal clinical trial, and I'm not making any recommendations on for treatment of particular cancers. So in terms of the disclosure that is potentially relevant to this is research support from the following pharmaceutical companies, which are primarily to support translational studies associated with clinical trials. This is a picture of me, and most of the a lot of stuff got said about me, so no more needs to be said about that. I also want to mention here Patrick Fitzgibbons, who was the co-chair of the Cancer Biomarker Reporting Committee with me that produced the Cancer Biomarker Reporting protocols. So let's jump into the actual lecture now, and the first thing I want to do is sort of set the problem for why these templates were created. So kind of part 1 of this lecture are the problems with reporting biomarkers in anatomic pathology. In most of the cancer protocols, the CAP, and when I say CAP, I don't mean the Canadian Association of Pathologists, but rather the College of American Pathologists when I'm referring to the protocols. There were multiple issues that were of concern. There was always a section in the list that were called ancillary studies, and sort of anything immunohistochemical or molecular sort of got thrown into this section, but there was no standardization of this at all, and we'll go into some more detail about this. There's also the recognition that with many of the ancillary studies, particularly things other than immunohistochemistry, there may be a delay between the time that the initial anatomic diagnosis is actually finalized and the report is the report's out, and when particularly the molecular results are available, which might be a week or more later. And so signing out the protocol at 2 different times was a lot of questions in the mind for people's minds for how these things should be reported. The reasons for the protocols was there was a lot of questions from cancer registries saying that they were having trouble analyzing data from molecular and biomarker studies because there were so many different ways of reporting the results, and it was very difficult for them to structure the data. So here's just an example for hormone receptor testing in breast cancer. These were surveys of 4 very large, well-respected national laboratories where we looked at various criteria for how they were reporting hormone receptor staining in breast cancer. And what we can see here is that they all report the percentage of staining, but then their reporting criteria and their cutoffs for what was constituted positive versus weakly positive, or various terminologies were somewhat different. The terminology was somewhat different. Some reported the fixative and the fixation time, which we know to be important for the results for this. Some of them didn't, and some of them told you what antibodies they were using, and some of them didn't. Really, the only thing that was consistent with these was the fact that they all reported the actual percentage of cancer nuclei that were positive. So it, it's just an example of the lack of uniformity for for reporting across for biomarkers. And then variation amongst the cancer protocols themselves. So breast cancer, because of the amount of work that's been done in this field with HER2 and with fairly cancer biomarker section, and so you can see that there was a fair amount of information that was solicited here. A lot of it was actually required because there actually are requirements for formal requirements for reporting cancer biomarkers in breast cancer. But then there were other sections, and here we chose gastric cancer as an example here, where it just says ancillary studies, you specify, and anything that you did, you would just kind of report here. And lots of the cancer protocols, the ancillary studies were sort of like this. It was just, well, if you did anything else, you can list it here. So in terms of the time delay between the initial anatomic diagnosis and the biomarkers, what you can see here's a protocol that's listed that sort of goes over a colon cancer here, and then you can see that often the ancillary testing section would just be listed as pending, and then might come on as a later addendum. So when the cancer biomarker reporting committee was first was first being formed, and we started out as a working group before we became a formal freestanding independent committee within the College of American Pathologists. The challenge was to develop standalone reporting templates for cancer biomarkers, both predictive and prognostic biomarkers, and these were intended to replace the quite variable ancillary study sections of the CAP cancer protocols. And the first three that were chosen because of the prevalence both of performing biomarker studies within these cancers, and also because of the amount of active research and the clinical use of biomarkers. The first three that we chose were breast, colon, and lung for templates, and then those were actually completed and published in late 2013. So into developing the templates, there were a series of expert panels that were created for each individual cancer type. There was a review of the evidence and recurrent recommendations from organizations such as CAP, ASCO, and NCCN. These panels were not trying to look at the evidence for doing these particular biomarkers. What we did was look at the data that was out there for the validation of biomarkers, and then there was also the consideration of how prevalent is a particular biomarker in clinical use, because the initial for these templates we wanted to address very common biomarkers. So there was a draft of a standardized reporting templates that were sort of modeled in the same way as the CAP cancer protocol, since they were sort of tended to marry up with the individual cancer protocols. And basically the structure of the templates is that you report the results and then the methods, and then there's some explanatory notes. We'll go some examples of these. So there was a lot of expert panel representation on these, because we wanted to involve all the potential stakeholders for these. So you can see a various number of professional organizations involving both medical oncologists and surgical oncologists, molecular pathologists. Cancer Care Ontario was also represented on the committees as well. So we did have some Canadian involvement. So these are the groups, and these groups were represented effectively on all of the committees that were producing these templates. So in terms of developing the templates, again, as we talked about, after the templates were created, there was expert review from the relevant scientific resource committees of the CAP. So this included, for instance, the Cancer Committee, looking at their protocol and then the proposed cancer biomarker template to make sure that they meshed and worked together well. The Molecular Oncology Committee also looked over these well to see if they had input, and in some cases the Immunohistochemistry and Surge Path committees looked at them as well. Also involved was the Pathology Electronic Reporting Committee, which is known as the PERT Committee within the CAP, and the CAP Structured Data Team, because these templates are intended to have electronic implementation as well, and we'll talk a little bit about that in a few minutes. There's been an open public comment period where these went up on the web, and people were able to say whatever they felt about them, and we got some really helpful comments and some of what I will refer to as humorous comments. Because no one likes, in the end, I suppose there's always some resistance to new guidelines or people or the thought that someone else may be telling us what to do. But overall the comments were favorable. And then once the comments were dealt with, these were then published. And during the question and answer period, we may actually go to the CAP website and actually look at and look at some of these, so I can actually show you where they're housed. And then there's the commitment going forward to maintaining and updating these with at least annual updates. Okay. So again, the goals here, we're trying to create these standardized structured reporting templates with gathering information and feedback from all the relevant stakeholders, publish these, and then try to make them electronically available as well. So these were the big goals of the committee. So here's an example of one of the templates here that's currently available for use. This is the one for non-small cell lung cancer. And here's the group of some of the people that were involved in this. I'm not sure if this is everybody. That may be cut off a little bit. But we're just going to talk here. I'll later go through in detail the melanoma template and talk about some of the evidence behind it and that. I just want to give an example here of lung sort of as a quick example. So you see here in the results section, EGFR mutational testing is an important biomarker for treatment in lung. So basically, the information required here is a mutation detected or is it not detected? What did you detect? And some of the common ones are listed here. If it doesn't happen to be one of the most common categories listed here, then there's a space here for one put in whatever one did find. And then there's some explanatory notes down below. There's in the methodology where you can list which of the common exons were actually assessed here, and then you can talk about the methodologies that you used. And again, this is select all that apply, because there will sometimes be say some groups might choose screen by next generation sequencing and then do some kind of confirmation by Sanger sequencing. So you have the ability to check off whatever whichever technology you use. This becomes important in some cases because some of the technologies actually have a different potential to select certain types of events, and we'll talk a little bit about that in the context of melanoma later. So the data elements within these reports right now, they could be required, but right now the only place where the biomarker template reporting is required, at least as far as the U.S. is concerned, is with the breast template, since there is actually required reporting elements for the breast biomarkers. For the rest of these, the reporting of biomarkers is optional. And then for all of these, the important thing is that reporting is conditional on the test being performed. So if the test wasn't performed, you need to put a lot of did not do, did not do, did not do. You only want to use the part of the template that corresponds to testing that you actually did perform. And again, as I think I mentioned earlier, these templates are not intended to make recommendations for what type of molecular or other ancillary testing is required for the various cancers. It's just for if you do perform a biomarker test, this is a way that one can report it. So again, here conditionally required items, you only report it if it's necessary. Here's the example of ER, where you're stating, is it positive or is it negative? And then you have some results for the intensity of staining. And then again, the things down here, bracketed in red, are required only if they are applicable. So the reporting responsibility, who's supposed to use the templates? The required data elements, again, are required only if testing for that particular marker has been done and the result is available to the pathologist. And then there's also the sense that an element is identified as being required for reporting purposes only and is not intended to mandate specific testing for individual patients. So again, these are not recommendations for testing; they're only recommendations for how to report testing once it has been performed. Okay, so kind of the ongoing challenges of this process is that some of these, because of the sheer amount of potential patients for the biomarker, made some of the templates rather lengthy. This is a very rapidly changing field, as I'm sure that you're all aware, and so the frequency of updates for this there's been a lot of debate around this as to how frequently these need to be updated. Biomarkers are increasingly being applied to many types of cancer beyond just the first three that we implement, and so trying to develop, all the new templates has been a challenge with the amount of manpower that we have. Making sure that there's harmonization so that the way that we're asking biomarkers to be reported in each of the cancer types is in a similar format. We feel is also important to make for ease of use with these. There was a lot of discussion about the differences between the ways that we test and report in solid and matter lymphoid neoplasms, and there's been some challenges here as well. And then question about what should be the format of the report. And then additional challenge has been how do we want to use, and this was something that was very important to the NIH and the CDC, is how we want to map the results to standardized terminologies such as LOINC and HGVS, which is the standard way that we report mutations. And so there was a lot of debate about these. So here's an example of some KRAS mutational pointings, trying to out how some of these templates became rather long. These are only for KRAS from colon. I believe this is from colon. Could also be from lung, but I think this one's colon. And so is a mutation there or is it not there? And then lots of listing of the common mutations here. These are not actually in HGVS. There was a lot of discussion about the formatting there, and for now at least we've decided to report them in the format that is most recognizable to the clinical teams that will be reporting and using this information. We'll talk a little bit more about that later. But you can see how some of these templates can become quite long. This is just one biomarker, almost a whole page. Here's an example of how the report might actually look. So for lung, there's a comment on specimen adequacy. tumor cellularity, which helps you determine what the potential sensitivity and avoiding false negatives. There's results listed for the various types of mutational analysis that were done in this case. Methodology, what was assessed, and how was it assessed? Here with the exons that were sequenced for EFR, ALK, and KRAS. And for this one, it says exons assessed, and if it was exons assessed, it really should have been 2, 3, 4. This is actually should read codons assessed. There's a mistake there. Anyway, so here's reporting basically global comments about the adequacy of the specimen. What are the results, and then how did you get those results? Basically, is the flow of the information as the reports actually create. So these are the templates that are currently in production and being worked on now. Some of these, such as gastrointestinal stromal tumor, gastric HER2, and some of the lymphoid ones have gone through public comment and are actually about to be published onto the web soon. In fact, when we go over there, they might even be on the web, but maybe not quite. Just as very close, I know. And then melanoma is another one, and that one I just have it in red and underlined because we're going to discuss it in some detail in the second half of this talk. So what are these biomarker templates? They're fully optional which is the example of breast, which does
Enterprise Express System to your user ID.
They are recommendations for how to report the results of biomarker performing, not recommendations for what tests should be performed. You only need to report these when you actually have the information itself. So you don't need to report them as pending, for instance. And they're recommended for the laboratories that are performing the test. So if
enter case number followed by the
there's a out for the test to another laboratory, it would be recommended that they would report in this type of format. These are to help standardize biomarker reporting. So what we don't intend these to be used for, they're not requirements. They're not requirements for using the templates at this point. That could change in the future. They are not asking the laboratories to report all of the elements at once. They can be used as the information becomes available. There are certainly not recommendations for what testing should be ordered. There are lots of other organizations and sets of validated recommendations for that available elsewhere, and not currently acquired for the accreditation process in the U.S. They're required for laboratories referring biomarker testing to a reference lab. So not asking the laboratories to report on the biomarkers if they're not currently doing so. There is, however, the recommendation that if you do receive results from the outside, that it would be reasonable to take those results and reformat them into an addendum in the surgical pathology report so that they're all available within the medical record in a structured form. Some of the ongoing issues and challenges is trying to create a standardized terminology for both paper and electronic versions, and this seems like it would be really easy. You just produce the paper version and then you make an electronic version. It's turned out to be a lot more complicated in terms of mapping these terminologies and what's required in order to keep that in line. And we're working with a number of other organizations, including the National Library of Medicine, the CDC, and the National Cancer Institute in the U.S. To develop these appropriate coding solutions, because they want to be able to use this information within their registries. So here's an example of the different reporting outputs where we talked about that this is a common mutation in EGFR, which is this P790M mutation, which is an activating mutation in EGFR. Here's a common way to report this in terms of clinical use, which tells you the gene, what exon it's in, and what the event is. The GVS, which is a particular non-ambiguous way to report a mutation, you give the reference gene or the reference sequence from the gene that you're reporting on, and then you give within that reference gene what is the change that occurred and where did it occur, and then what does this look like at the protein level. And there's also ways that you can do this by genomic locus as well, which you just talk about specifically where the event is within the entire human genome. This last method is the most complicated way to do this, and is probably not what's going to be recommended. I think with time we'll probably evolve toward reporting these in both a quote-unquote human readable form, and then also with the HGBS format, which is an unambiguous way for registries to interpret the mutational data. So recently the CDC, in fact, CAP was awarded a large grant from this in order to help study and develop the biomarkers, and this will actually be implemented on a small scale to test and see how well these can be applied with a who's actually producing the results, and then how that translates into their interpretation within a cancer registry. So again, as you probably know, there are electronic cancer checklist packages for the other cancer protocols, and the process is underway now of doing the same thing with the biomarker checklists as well. And so these, theoretically, the electronic version of the protocols and the templates would be available for integration into the laboratory information system. And currently, the breast, colorectal, and lung biomarker templates do in fact have an ECC version, which is basically driven by a menu where you actually just click the results, and then based on the logic of what you click, certain options will disappear or appear, which was somewhat helpful for simplicity of use. So again, these are smart templates that assist for the workflow with, again, as I was talking about, the conditional branching logic. And then these e-forms available from CAP and from a variety now of different vendors should be helpful in applying the cancer biomarkers in terms of making them. You should be able to automatically update these when new template versions are released, and hopefully they fit nicely into the individual pathologist workflow. A thing that can occur so this is after the public comment period, while the protocols are being used. There's actually an area that solicits user-submitted issues, both for the electronic and the paper format of the protocol. So if people have, if something is unclear, or they're not sure how to interpret something, or they think that there should be more clear, or other things that should be included in the protocol, all of that information can be entered. We keep track of that, and then that's discussed on a regular basis to see whether we want to update that with the next release. And then if it's something that can be answered quickly from one of the experts on the expert panels, usually the person who's chairing the individual expert panel, an email addressing the individual user's question will go out, either telling them we're looking at this and we may use it, or hey, here's the answer to what you're asking. And this is just where the cancer protocols are located right now on the CAP site, and the ones that are up for public comment are also in this area as well. Okay, so we move on now to sort of the second part of the lecture, where we're going to look more closely at the melanoma template and talk about why. We're going to sort of go through the whole template kind of in detail, and then we're going to talk about why we're reporting what we're reporting in there. So kind of give some background and some context for this. So now for the second part of the lecture, here's kind of our objectives. So we're going to introduce the proposed melanoma biomarker template in detail. So the proposed biomarker template, the stage that it's at now, is that it has gone through its public comment, gotten the public comment back. we've addressed the public comment, and it's now in the process of being converted into its electronic format, which will then test the logic within that and make sure that it makes sense. So that's kind of where the melanoma biomarker template is at this point. So the other thing I want to do is talk about some of the molecular oncology of melanoma and why we have certain elements within the template and why other elements were not included within the template. So this includes some clinical context, some of the particular molecular derangements, a little bit of detail about some of the methods of detection, and then some anatomic pathology considerations as well. So this is the CAP, one of the versions of the CAP melanoma biomarker report. This is the group of people that worked on it here. And to step back now, I want to give you just a little bit of information about what's going on genetically at a very high level with melanoma, and then we'll focus down on particular biomarkers. So most of you are probably aware that there's a large project that's sponsored by the National Cancer Institute in the U.S. There's been a lot of Canadian participation in this, the Cancer Genome Atlas, where we're sequencing the entire genomes of a variety of different cancer types. And one of the things that's going to be unique about melanoma is it has an extremely high mutational load, so it's one of the most heavily mutated of all the human cancers, and that there's particular types of mutations within melanoma, as shown in the yellow box here, which is the C to T transition. And what this C to T transition does is it's a marker of UV exposure, which makes a lot of sense for melanoma. Now, melanoma as a whole, melanoma is one of the depending on how you measure it, one of the very highest mutated cancers. The world leaders in mutational load for cancers are anything that has microsatellite instability. So the subsets of, say, colorectal and uterine carcinoma that show microsatellite instability have just huge numbers of mutations within them. But melanoma is still certainly one of the highest. And what you can see is that the cancers that tend to be higher are those that are associated with some type of environmental or external assault. Those are the ones that tend to have the higher mutational loads. There's lots of different types of mutations that can occur, though. The vast majority of mutations that occur in most types of cancer are single base pair substitutions, shown in blue. But you also see deletions, you can see amplifications, translocations, and other types of events as well. But within most cancer types, those are relatively rare events compared to the single base pair substitutions. So in terms of now going down to the level of the actual gene that's mutated, there's kind of two broad classes of genes that we think about in cancer, and those are the cancer oncogenes. And with oncogenes, that's a gene that gets turned on permanently, and so it is then, as opposed to being turned on and off in a regular fashion, it's just on all the time. And to turn a gene on all the time, there's only, for most proteins, only a few ways that you can do that. And so with oncogenes, you tend to have very patterned mutations in many cases that occur only at particular sites, which are sites that are, that are the ones that allow you to augment and leave the on switch on all the time. The second class of cancer genes are so-called tumor suppressor genes. And tumor suppressor gene, what you're trying to do there is destroy the function of the gene so that the protein's no longer functional. And if you're trying to break something, there are a lot more ways to break something than there is to make it stay on all the time. And so within you tend to have mutations that can occur throughout the open reading frame of the gene, and they're sort of scattered all over, and many of these result in early stoppage of translation of the protein. So while there's lots of different genes that can be mutated in lots of different pathways, and now we're developing all kinds of different clinically available inhibitors for these, so that the field is somewhat complicated. However, if you think and take the big picture, there are some relatively simple, very broad pathways, both in terms of biochemical cascades and also function, that you can sort of classify each of these genes into, which somewhat simplifies the field. So with that sort of background for melanoma, let's now skip into, melanoma has literally in most hundreds to thousands of mutations across its genome, but there's actually only a few of those that are really important, that actually drive the development of melanoma. And it's not clear that we understand necessarily what all of those are, but we're starting to get a good idea. So, if you think about in general what melanoma needs to do, there's two really important pathways. There are other important pathways as well, but for the types of biomarkers that we're going to talk about with clinical importance, the main players are depicted here. So you have the BRAF MEK-ERK CDK pathway, which is generally associated with proliferation, and you have the PI3 kinase AKT pathway that's generally associated with survival. And for melanoma in general, what you want to do is actually activate both of those pathways, okay? And this next slide shows us some of the ways that melanoma cells actually do that. So this is just a different way of looking at the pathway. So as most of you know, BRAF mutation is relatively common in certain subtypes of melanoma, a little bit less than half the cases. So that activates this pathway here. NRAS, 20% of melanomas, it activates both this ERK pathway. It also goes over and tickles P3 kinase and activates this AKT pathway as well. P10 has aberrations, so this would be actually loss of function. So this would be a tumor suppressor gene, whereas these two are oncogenes. Is lost in a significant number of cases, and this, interestingly, this loss tends to be combined with BRAF mutations, so that you activation both of the ERK pathway and also the AKT pathway. And then the other thing which this slide doesn't show is that you can have activations in upstream pathways such as the KIT proto-oncogene that actually activates both of these pathways as well. Okay, so the story is a little bit more complex than that, because I've been talking about melanoma as if melanoma is a monolithic disease, and in fact it's not. There's different types of melanomas, and these different types of melanomas have different types of mutations in them. So if we look at BRAF and NRAS, you can see here with the cutaneous melanoma with intermittent sun damage, you have pretty high frequencies of BRAF and NRAS mutations. But for these acral melanoma, mucosal melanomas, or melanomas that are associated with lots and lots of sun damage, lower rates of BRAF and NRAS mutations. And then in uveal melanoma, which is melanoma of the eye, you basically don't see either BRAF or NRAS mutations. Look at KIT. KIT mutations not seen really in cutaneous melanoma or in uveal melanoma, but seen at various prevalences within these acral and mucosal melanomas. And then finally, there's the G protein alpha subunit homologs GNAQ and GNA11, which are mutated at very high frequency, virtually all cases of uveal melanoma, but more or less not seen within these other types of melanomas. So you can see that the mutational group, it really is not a monolithic disease. The different clinical subsets of melanoma have different molecular biomarker correlates associated with them as well. So let's talk now here a little bit more about BRAF. And what you can see here is that this is the initial description from 2002 of of the BRAF oncogene from the group of Andy Futriol, whose lab is now right next door to mine. And so he was the first person to show that BRAF was mutated in multiple different types of human cancer, and then it became pretty quickly apparent that it had a really high prevalence in melanoma. So again, we said about 45% of non-sun-damaged melanoma, a little bit lower in acral and mucosal and not seen in uveal. And the majority of these affect a single site within BRAF, which is this V600 site. A lot of it is V600E or valine to glutamine, but there's also a subset of cases that are valine to lysine, and then there's about 10% of cases where valine is mutated to a variety of different things. And then there are a few other mutations within exon 15, and then there are some exon 11 mutations in this as well. Also, other alterations seen in melanoma, which we won't talk as much about. They're less common. You can actually have amplification or increased copy number of the BRAF gene, and you can see translocation or gene fusions that drive overproduction of BRAF as well. And these actually potentially have different treatment implications. So if we look at all the different mutations that can occur within BRAF, and this is sort of looking at the amount of catalytic activity versus wild-type BRAF, you can see that there are some mutants that produce really strong activation, and other ones that are not that much more active than wild-type is, and it sort of varies. But the really strong activators are in this V600 area. So if we look at BRAF in terms of clinical outcome, there may actually be some difference in terms of clinical outcome depending on whether V600E versus V600K is the actual mutation. There's also differences in terms of the sex distribution of melanoma based on whether you have V600E or V600K, and also the site of melanoma, which are interesting. And this is some work shown from a couple of there's data from Australia as well. Now, the really important thing, though, but we don't really use these differences in mutational status and the potential differences in outcome to actually manage melanoma. It's just an interesting biological feature. But what's the interesting thing that you all know about BRAF mutation is that there are now, based on the presence of V600BRAF mutations, that there are multiple FDA-approved treatments for this, and all of these are inhibitors either of BRAF itself or of MEK, which is a downstream of the BRAF activation cascade. So, in general, BRAF mutations that are not V600 do not tend to be responsive to the V600 selective BRAF inhibitors such as vimorafenib and dabrafenib. Trametinib is the MEK inhibitor, some of these do respond to the MEK inhibitors, and there's a lot of study of looking at whether you should potentially be inhibiting BRAF using a combination of both BRAF and MEK inhibitors, and there's some interesting mechanistic reasons for that, which we don't have time to go into. Just an example of a phase 1 trial with trametinib, MEK inhibitor, for inhibiting BRAF melanoma, and you can see here that with this waterfall plot that a significant number of patients actually respond. And here's a case of a patient here after a few cycles of therapies that showed a rather dramatic response. Now the problem, as you probably also know, with the inhibition of BRAF is that resistance often ensues with this. But the fact that we've got metastatic stage 4 melanoma responding to any type of therapy is quite remarkable, and it has really transformed the oncologic care of metastatic melanoma. The thing that's really important, and part of the reason why these biomarkers are important, is that if a patient doesn't have a BRAF mutation and you give them a BRAF inhibitor, they don't have any benefit from that. And of course, the drug itself is very expensive. But even worse than that, it looks like that if you give a BRAF inhibitor to a patient who doesn't have a BRAF mutation, it may actually accelerate the course of the disease. So not only have you given them something that's really expensive and won't help them, it may be really expensive and potentially actually speed up the progression of their disease. So the proper reporting and testing for these biomarkers is absolutely critically important for the management of melanoma. Let's move on to one of the other oncogenes now that's important for melanoma. That's NRAS. Again, it occurs at a variety of different levels depending on the melanoma. NRAS itself, particularly now that we have the ability to inhibit BRAF as inhibitors, kind of marks for melanoma that has a worse outcome. This is a study from the Sydney Melanoma group. This is a study showing similar results from our group. These are patients that for a long time we've struggled to try to figure out how to treat, but we'll talk about some emerging treatments, for these. In fact, we'll do that right here. So, very recently, we haven't really known how to treat NRAS mutant melanoma. Inhibitors for RAS itself have not been particularly clinically successful, but a bioinformatic approach with some experimental validation from Linda Chin's group, who's at our institution, has recently shown that a combination of a MEK inhibitor and a CDK4 inhibitor seems to show some efficacy for inhibiting RAS within RAS mutated melanoma. This was an interesting idea and is sort of counterintuitive because again you're inhibiting the same pathway at two different levels. And so this was kind of a surprising result. I don't have time to go into the bioinformatics as to how this was discovered, but very rapidly the idea was taken and was tried in terms of a clinical trial because there were actually CDK4 inhibitors that were clinically available that are used in other cancer types. So this is a clinical trial that's looking at the combination of CDK4 inhibitor and a MEK inhibitor. This is data that was presented at ASCO. And so the combination here is a MEK inhibitor and then a CDK4 inhibitor. And what we can see here with NRAS mutated melanoma is again from the waterfall plots, a fairly remarkable response to both of these agents from melanoma patients. So now knowing that one has an NRAS mutation may actually be important for how melanoma is going to be treated. Again, these are clinical trials. This is not an FDA approved treatment, but these are FDA approved and sponsored clinical trials. Another interesting view of using CDK4 as a target in melanoma may be that CDK2 loss itself may also help be predictive of whether patients are going to respond to CDK4 inhibitors. And we'll address that a little bit more right at the end of this lecture. So the specific mutations that we're particularly interested in melanoma in terms of things that are very common clinically are KIT mutations, NRAS mutations, and BRAF mutations. And just quickly, some data from KIT mutations. Here's one of the early published cases of the Journal of Clinical Oncology showing treatment of a KIT mutated melanoma with a KIT inhibitor and showing the dramatic response on KIT. And here is a clinical trial look at a inhibitor for KIT in KIT mutated melanomas, showing that efficacy was seen within this population. And there was also the suggestion that KIT mutation with additional amplification of the KIT locus might be the best predictor of who was going to respond to this drug. Again, this is the drug that was used in this case is FDA approved, but it does not have an indication in melanoma. So this is a clinical trial and not an FDA approved treatment, although the drug itself is FDA approved. Okay, so with that kind of background for what's important in terms of biomarkers in melanoma, and at this point for clinical management, it's BRAF, NRAS, and KIT. Let's go in detail through the initial draft of the melanoma template. One of the ways we can actually look for the V600E mutation is for looking for the mutated protein. It's actually an antibody that's specific for this. That would then indicate that the patient has a V600E mutation. Here's just an example of a melanoma that's wild type versus a melanoma that has a mutation here demonstrated by next generation sequencing, and you can see here the nice strong recognition by this antibody of the V600 mutation. Types of mutations at V600 don't actually work, so this antibody will miss other types of mutations that occur at the 600 position. But people like to use this, and so we create a report, section of the report for this is determined whether, basically reporting whether immunohistochemistry with this is positive or negative. Again, the report doesn't address for the FDA-approved treatments with BRAF inhibitors for melanoma. There are also FDA-approved companion diagnostics, and V600E immunohistochemistry is not one of those, although there are lots of people within the U.S. that are not using the particular companion diagnostics that were approved for BRAF testing. So in terms of looking at BRAF mutational analysis, you've got negative or you've got mutation detected, and then you've got a listing here of some of the most common mutations that are present within melanoma, and then the ability to just click off which one you see there. And again, just as I was telling you, it's these mutations at V600 that are by far the most common in melanoma, and they tend to occur primarily as V600E or V600K. This is just detection of this by next generation sequencing. Now, in terms of techniques for sequencing, different techniques that can be used. With Sanger sequencing, which kind of detects all variants within the area that you sequence, it's a little slower and more time-consuming. Iris sequencing has been used, and it's really good for looking at hotspot mutations, such as in BRAF and NRAS, where the mutations are at specific sites. It's faster, it's more semi-quantitative. The first sort of multiplexed way to look at mutations, where you could look at multiple different genes at the same time, was a mass spec based technology. This is not all that commonly used anymore. So then within BRAF, once we've reported what results are, you then want to list the types of what technology you use. So this particular first test is the companion diagnostic for that's FDA approved for dabrafenib. This is actually an older version of this. There's actually now the second thing listed on this is the FDA approved diagnostic for the other BRAF inhibitor as well. So the actual template now has changed a little bit from this example that I'm showing you. And then here's just lots of other ways that you can use potentially to detect the mutation. Or if you happen to use none of these, then here, you can put down what you did actually use. Here's the difference in terms of what the different data looks like from the different outputs of different methodologies, from Sanger sequencing of multiple different flavors of the BRAF mutation to sequencing to sequenome. Show you what the different technologies, the output looks like. So NRAS again, and now have a much bigger selection because the number of prevalent mutations in NRAS in melanoma is a little bit broader spectrum than with BRAF. So you've got, do you have a mutation, yes or no, and then if you do have a mutation, what is it? And then here's your list of things to check off here, or to just report one of these basically. And again, this sort of corresponds to the different sites and flavors of where the mutations are within melanoma in NRAS. Just an example of detecting an NRAS mutation by next-gen sequencing. And then again here, there's not an FDA-approved companion diagnostic for NRAS because there's not an FDA-approved treatment at this point for NRAS-mutated melanoma. So we just listed the methods that are most commonly used here in terms of selecting the method. With KIT, again, you've got do you have a KIT mutation? Yes. No or yes, and then if you do have a KIT mutation, these are listing here some of the most common KIT mutations, although as I'm about to show you, there's a lot of different ways you can have KIT mutated. KIT mutations in melanoma show a different distribution than what we see in gastrointestinal stromal tumor. They tend to be point mutations, and they tend to be not involve exon 9 and have a lot more involvement of exons 13 and 17 than untreated gastrointestinal stromal tumor does. So here's sort of a listing of the from a few years in the literature of what the most prevalent types of mutations are in terms of the point mutations involving the different exons within for melanoma. And here there's not a companion diagnostic, an approved companion diagnostic for this. So again, we've just listed the various methodologies that could be used to assess KIT mutations. And the reason that we've listed so many different methods here is that there's a lot of evolution, within different ways using different technology. This is just an example from our own molecular diagnostics laboratory at MD Anderson, showing before April 2012, we had all kinds of different techniques that we were using to detect mutations in different genes. And then with time, we've increasingly started using next-generation sequencing as the primary workhorse within our laboratory, which has sort of changed the distribution of the different types of testing that we're doing in terms of the percentage distribution, so that most of our work now is done using next-generation sequencing. So that's why I've offered all of these different methodologies for people to choose which one they're actually using. So those initial biomarkers that we chose as the most important right now in melanoma that are being done very commonly. We realize that there may be other things that people are reporting, so we do have a section within here to report other types of markers and results. So as you know, from all these sequencing projects of melanoma and other cancers, we're now getting a lot of idea of what the additional mutations may be. A play that's probably going to end up being important in melanoma are these loss of function mutations in the neurofibromatosis type 1 gene, which is another way of activating the RAS and probably also the PI3 kinase pathway, and these occur at a relatively high percentage within melanoma, and this may eventually decide treatments based on this. But this is research work right now, and it's very, very theoretical, and so these aren't the kind of genes that we would include on the template. I'm just showing you as an example that we know that the field is going to change. Whether or not P10 is mutated may determine whether or not you might want to use an AKT or a PI3 kinase inhibitor. I showed you data here earlier about what type of inhibitor you may want to use if you have a deletion or mutation in CDKN2A, and then you can see that these are rather prevalent within melanoma, and that may make a difference as well. But all this is sort of emerging, and so we decided not to include those at the present time within the template, and that the template should only be very well-validated biomarkers. Although I think with time, some of these other events are clearly going to move into the template. The other thing that we didn't choose to stick in the template, which I showed you that very commonly these melanomas, uveal melanomas, here with the pigment and here bleach, so you can see the nuclei, to have these mutations in GNAQ or GNA11. We don't have a clinical use for those right now, and so we didn't really address these mutations. If they become more clinically useful in the future and are actually used for particular clinical decisions, then we would include them in the template, but we decided not to do that at this point. So the important thing about cancer treatment now, at least for metastatic melanoma, for many cancer types, is by doing these types of biomarker or molecular profiling, we're showing that they're, in our particular case that we just talked about, melanomas that have lots of different molecular profiles, and those molecular profiles are going to suggest that different treatments may be more successful. And what's important is that we're able to deliver the results of these biomarker testing to our clinical colleagues and also for cancer registries in very clear fashion. And that's sort of ultimately what the biomarker reporting templates are for. Just summarize this melanoma section. Metastatic melanoma has kind of gone from untreatable to now having multiple options. We didn't even talk about the therapy revolution in melanoma, and ultimately the melanoma template may include lots of immunological biomarkers in it, which are likely to end up being predictive of whether melanoma is going to respond to PD-1, PD-L1, or other types of immune modulatory treatments, and so that may be part of the future of the melanoma template as well. Melanoma data is now certainly required to choose the optimal treatment in melanoma. We certainly need to have clear communication with our colleagues, and the type of data on biomarkers that we're producing need to be tracked by cancer registries and the CDC in order to do their population studies and also quality metrics. As a pathology imperative, understanding and clear reporting of these biomarkers certainly creates credibility with our oncology colleagues. We certainly need to understand this, and as pathologists, we really need to be at the forefront of molecular oncology testing, as this now determines treatment in many cases once the cancer becomes metastatic, even more so than does the histologic diagnosis. Acknowledgements from the lectures. I acknowledge Patrick, the co-chair earlier of the Cancer Biomarker Reporting Group, and then I have a very important research collaborator that I work on with melanoma and melanoma medical oncology at my institution, Mike Davies. So thank you very much for your participation. I guess we can do questions now if anybody's still on the line.
There is time allotted to questions. I'd like to remind everybody to use the chat function if you have any questions for Dr. Lazar. Okay. So I did have a question with regarding the future of the biomarker reporting. As you alluded to at your institution, a lot of the molecular testing has moved into next generation sequencing. That's certainly the trend that we're seeing here in Ontario. The current model of reporting biomarkers works very well for the individual genes. When you're talking about panels or even whole genome sequencing, you can see the current reporting structure become wieldy very quickly. You all of a sudden start talking about reporting findings on a dozen different genes. Do you know how the checklists are planning to evolve to handle things such as panel-based testing?
Yeah, so I'll just admit to you up front, since we do virtually all of our testing with large molecular testing with large panels, I've actually clinically never used one of these templates because by the time they were developed, we were doing panels of 50 to 400 genes. And these really aren't applicable to that type of testing. The way that we're thinking about reporting and discussing next generation sequencing is evolving. In terms of what the College of American Pathologists is doing, we've been much more focused on trying to figure out how to do proficiency testing for next generation sequencing because of some of the ways that the FDA is thinking about regulating this type of testing within the U.S. And there's not a lot of consensus yet about actually how to report the findings in next generation sequencing. So I think in general the attitude at this point is let's let the field evolve a little bit more and kind of see how people are actually doing their reporting, and try to figure out what we can do in terms of guidelines for that. Because at this point, everybody was sort of a little reticent to try to produce guidelines for how to do reporting in a field that's evolving as rapidly as the panel-based next generation sequencing is doing. So that's kind of a cop-out answer, but it's the most honest answer I can give.
No, I appreciate that. Trying to wrap our heads around how to accurately report on these large panels has been very difficult, especially when we try to look at solutions on a provincial basis.
I think there are principles emerging with this, and I think that most people that are doing this agree that when you're doing a very large testing of a panel of genes, that you probably want to list the things that are actually positive up very high in the report and very prominently, and that you may want to elevate some really pertinent negatives based on, what type of cancer is being tested as well. For sure, you want to do the things that are positive, and then, but the way that people have decided to parse how they want to report the genes in terms of is this clinically actionable or is this not clinically actionable, a lot of that is really interpretive at this point. And so providing recommendations for that is kind of tough, and it sort of starts across the line of making treatment recommendations. It sort of blends treatment recommendations with reporting recommendations, which is want to get into the treatment recommendation business within these cancer reporting groups.
There's a question that came in through the chat, and that is, who is responsible for reporting the molecular results? Surgical pathologist or the molecular lab directly?
Okay, well, I think you can look at that in multiple ways, and there are successful models of both. I think. One place we're neutral as to that. What we're suggesting is that whoever reports it, they should really think about including the elements that we're talking about here. And there's also the thought that maybe molecular testing needs to have the same type of synoptic reporting that we're all evolving toward with surgical pathology. So in some cases, the molecular diagnostics, the testing will be done by the molecular lab, and then the results will be added as an addendum onto a surgical pathology report, and then the individual surgical pathologist might make some comments about the molecular results as they're relevant to the particular histologic features of the surgical case, and then send that out, and then it's an addendum there. In other cases, the molecular diagnostics group, may produce either an addendum or a separate but linked report, that reports the molecular diagnostics, directly. Like I said, the templates themselves are agnostic as to who should do the reporting. For me, I actually do both. I go back and forth between sending out my surgical pathology cases, on the days that I do that each month, and then and then signing out the other side. At our place, the nation sequencing is actually signed out as a separate but linked report. For instance, however, when I sign out sarcomas, the molecular testing that I do there to confirm the diagnosis with a translocation, I actually take and put it in the tail end of my report because it's very relevant to the particular diagnostic features that I've talked about in the report. So, that's a couple of, I don't know, it's probably more information than you wanted about it. I think basically you just need to have a good system that clearly reports it and makes sure that the communication with your oncology colleagues that they understand, have access to, and know when the information is there and ready for them to access and use to make their decisions clinically.
Okay. But the Ontario note on that, starting in April of this year, we have mandated the reporting for colon, lung, and breast, and the filling out of the synoptic, the check with synoptic is a responsibility of the lab that does the biomarker testing, and whether that's the staff in the molecular lab, a surgical pathologist, doesn't matter, but the reporting is done by whoever does the testing in Ontario. Now,
that certainly makes sense, and in general so I should make that clear because I actually addressed that within the lecture. The cover is initially the biomarker templates initially, we argue, are applicable to the people who are signing the initial reporting of these. Depending on whether your testing is done in-house or outside, an outside group may do it, and in that case, you may want to, re-report those. But yeah, no, it's the bias with ours would be whoever signs out the initial report. That this should be the format that's used. Yeah, it's actually interesting that in the Canadian provinces usually tend to implement and discover and do a lot of the early discovery of use of the templates, and we get a lot of great feedback from you guys because you guys tend to implement them much more rapidly and widely than we do initially. So we actually always learn a lot from your experience.
I'm going to paraphrase the next question, and it has to do with molecular testing. Whether it's better to test metastatic disease or the primary tumor.
Okay. So, in general, all things being equal, and the samples from both sites were equally good. You want to test the most recent. You want to test the most recently available metastatic disease, because that's the closest thing to what your colleagues are going to be treating with systemic chemotherapy or targeted therapy. However, depending on the cancer type and the literature, the degree of concordance in terms of molecular testing between primaries and METs can be very high. So, for instance, in melanoma, the testing that we've done to look at both primaries and metastases, the rate of concordance between BRAF, NRAS, KIT, P53 were the main ones that we looked at. And this was across some varied data sets were extraordinarily high. They were between 95 and 100% concordant. And so while we would prefer to test the most recent metastatic disease for the reasons that I told you, we know that if that's not available for some reason, or the sample's bad, or that, we don't mind testing the primary disease. But in general, you want to test the most recent metastatic disease.
Do you actually going after the metastases for molecular testing?
No, not unless I wouldn't necessarily. I would start with testing whatever you have, because depending on where the metastatic disease is, it always sounds real easy. Oh yeah, go get another piece of it. But if that piece happens to be a metastasis to the lung, every time you do, a CT guided biopsy, there you have a percentage of times where you're going to collapse the person's lung, no matter how good your interventional radiologists are, and so the amount of data that you can. I would suggest that you would test whatever's available, and then if for some reason there's a complete discordance between what your results are clinically based on what the mutational profile that you have, then you might want to go after, and try to get another piece to see if that could direct your treatment better. That's an evolving field, and my recommendation for that, change a year from now based on additional data that becomes available.
Okay. All right.
And the other little nasty secret is that in certain types, particularly with resistance emerging, for instance, a lot of studies been done in gastrointestinal stromal tumor, which was one of the early tumors where targeted therapy was used. So there you have KIT mutations. You use first line therapy for that is usually imatinib mesylate, but then the problem is after some months to use, resistance emerges, and then you'll often have reactivation of multifocal disease that becomes KIT positive and starts growing, and then unfortunately what was found was that as you tested different metastatic sites there, they tended to exhibit different mechanisms of resistance, each of which might or might not respond to particular therapies. And so what you would test becomes very complicated, and in many cases would be predictive only of what that single metastatic site would respond to versus all sites of disease. That's a particular complex question, but it just shows you some of the complexity about with solid tumors. That's the great advantage that the heme path and the bloodborne tumors have, is that their mutational load and their tumor all gets mixed in the bloodstream. It's really easy to take a biopsy, do some sorting, and look at the entire mutational load of the tumor, from taking a sample of sorted peripheral blood. But we don't have those advantages right now in solid tumors.
Okay, I think that's all the questions that have come through our chat line, and we're just approaching 2:30. On behalf of Cancer Care Ontario, the partnership, and the Canadian Association of Pathologists, I'd like to thank Dr. Lazar for a truly comprehensive, informative, and very clear presentation today. For the first session of 2015 series of CAP checklist presentations, we would welcome your comments and suggestions for ways to ensure these sessions are informative and relevant to your practice. Your feedback and suggestions as part of your completed online evaluations. WebEx recording of this presentation becomes available. Made available for distribution via links posted by Cancer Care Ontario, SAC, and the Canadian Association of Pathologists. This recording will be available for review at your convenience and not restricted. As a reminder, both the live and recorded presentations are eligible for CME credits. Please see session notice for more information. Certificates for each CAP checklist education sessions will only be issued for 1 month from the presentation date. The sessions will remain available electronically for an undetermined period of time, but the certificates will only be issued for the 1 month. So please refer to the session notes for the exact deadline date. To remind everybody on the call that the next education session is scheduled for January 28th, and I will be discussing thymoma and mesothelioma with presenters of Dr. Andrew Churg and David Wang. Thank everybody for attending, and again, thank you very much to Dr. Lazar.
Thank you much. Thank you. Thanks everybody for listening.
Bye.